Alex, what are you working on?
I have a debate in my next class that is entirely about these unusual cells and how they can affect medicine. I have absolutely no idea how I'm going to talk about how a stem cell works, let alone take a stance in the debate. Aaron, I don't think that I can do this.
Calm down. It'll be okay. I actually just wrote a paper about stem cells, so I may be able to help explain any questions you may have about the subject. It looks like you have already been doing some research. What have you learned so far about stem cells?
I've learned that they're undifferentiated cells, whatever that means.
Okay, well that is true. They are called undifferentiated because the cells aren't fated to become a particular type of tissue yet. To be a little more technical, we would say these cells have not been determined, so they have the potential to turn into any cell found in the body. Early in development, cells gradually become specified to fall down a particular cell line, such as digestive cells or nervous system cells. Eventually, these cells are said to be determined. The cells will now progress towards becoming a certain tissue no matter their environment. Genetically, stem cells have a fully active genome. In practicality, this means that stem cells have the ability to express any gene in the genome of the organism. They can express brain genes, heart genes, liver genes, whatever is needed. However, efficiency overrules versatility. During development, genes become inactivated, usually by methylation. This leaves the cell with only certain genes activated to work as a particular kind of tissue. It's more efficient if your heart spends all of its resources only making heart proteins and not wasting resources on brain proteins.
So that's how scientists are able to use stem cells to create new tissue in the body? That's actually pretty cool. But this book here says that there are different types of stem cells. How am I supposed to know the difference between amniotic and induced pluripotent stem cells, for example?
There are actually 4 different types of stem cells. The first type of stem cells we should talk about are embryonic stem cells because those are the best understood. These stem cells are only found in the developing embryo of an organism. To harvest embryonic stem cells, scientists have to acquire a fertilized embryo for growth, since early-stage embryos are made almost entirely of stem cells. The embryonic cells are then placed in a culture and refrigerated before they can be experimented on. The methods of acquiring this embryo are where the main ethical arguments are present in the debate about stem cell research.
Well, if they're so heavily controversial, then why use them? My book says there are plenty of other types of stem cells to use in research.
Well, yes, there are other types of stem cells that scientists could use, but embryonic cells have the potential to differentiate into any type of cell in the body and have a much longer shelf life compared to the other types of stem cells. Embryonic stem cells have almost no epigenetic inactivation, and cells with all their genes turned on are very useful for creating virtually any type of tissue. In addition, research conducted with embryonic stem cells usually use the leftover embryos from in vitro pregnancies that would otherwise be discarded or destroyed. These cells are currently harvested with the consent of donors and can be researched to find out what triggers the stem cells to turn into specific somatic cells.
Oh, okay. So what's another type of stem cell?
Kind of along the same lines as embryonic stem cells, there are stem cells that can be harvested from the amniotic fluid. Sometimes during the pregnancy, the mother undergoes a process called amniocentesis. This is where there is a sampling of the amniotic fluid to test for a range of genetic disorders that the fetus may have.
That sounds dangerous.
Well, there is some risk involved in the procedure of amniocentesis, but it remains a fairly common procedure, especially if there is a risk of a genetic disorder. The use of amniotic stem cells has an ethical advantage over embryonic stem cells because the embryo does not have to be terminated in order to harvest the cells. Amniotic stem cells also do not have the tendency to tumorize when injected into the body, which is a common occurrence in embryonic stem cells. Embryonic stem cells behave very nicely when in vitro. However, for some currently unknown reason, embryonic stem cells have a tendency to form cancerous tumors when grown in vivo. Amniotic stem cells do not have these issues, but they are not as potent or easy to work with as embryonic stem cells, which is why they are not used as frequently.
So what are these iPSC things I read about? Sounds like somebody spilt some alphabet soup.
Well, iPSCs stand for induced pluripotent stem cells. These are adult somatic cells that are injected with stem cell genes via a viral vector. In about 0.1% of cells, the introduction of these genes, called reprogramming factors, you can observe some cell colonies that have reverted back to the pluripotent stem cell state. Reprogramming factors are a series of genes cloned in vitro that are associated with undifferentiated cells. The hope is by injecting these genes into the cells will incorporate these activated genes into their genome, which allows them to transcribe genes they wouldn't normally transcribe. In essence, these give the cells more potency, hence the name induced pluripotent cells. This process tends to take 1 to 2 weeks.
That sounds like a lot of trouble. According to my research, it says that this type of stem cell has a lot of obstacles and a lower potency. What does that mean?
Before I explain potency, let me remind you about genomic equivalence. Basically, what that means is every somatic cell in your body has the exact same DNA except your immune cells, but I won't go into that. This means that your skin cells have the DNA code necessary to produce insulin, a hormone made by your pancreas. But skin cells certainly don't produce insulin. That's the job of the pancreas. This idea that all cells have potential to make proteins, but different types of cells make specific kinds of proteins, is where the idea of potency comes from. Potency is the power of a cell to become different types of tissue. Cells with the highest potency, what we would call totipotent cells, have very little epigenetic inactivation. This means almost all of their genes have the ability to be transcribed if the correct transcription factors are introduced. However, because cells have limited resources, most of their genomes undergo processes like methylation and acetylation to allow some genes to be actively transcribed within them, even though the genes are still present. These modifications are made for the reasons of efficiency, as I mentioned earlier, and the cells that undergo these processes are considered less potent. iPSCs have a lower potency than other stem cells simply because the process of reactivating genes in a fully developed cell is tricky, especially for researchers to do. The mechanisms that the body uses to reactivate genes are extremely sophisticated and hard to replicate in a lab. For example, the body uses processes like base excision repair to remove methylated cytosines from DNA. Making iPSCs is a shot in the dark. We shoot active stem cell genes into adult cells and hope that some of the cells will incorporate the stem cell genes into their genome. If they do, then there's a chance they will reacquire some potency they've lost during development. The researchers who discovered this process won a Nobel Prize for their work because their discovery was so amazing. A benefit to using iPSCs is that the cells come from the patient and won't be rejected by the immune system. However, there needs to be a lot more research in regards to iPSCs before we can be able to use them for a wider range of problems. As of right now, due to the lower potency, there isn't much that can be done with them medically, though researchers are very optimistic about iPSCs and their future.
So do adults have stem cells?
Yes, actually. Adults have somatic stem cells. Somatic stem cells present in an adult have narrow potencies. There have been studies about mesenchymal cells in bone marrow that can be used to produce new tissue, such as blood cells. But like iPSCs, due to the low potency, not much can be done to manipulate these cells, as they have already partially differentiated. They are really kept in the body to make new cells in the same line as themselves and replace worn-out cells. For example, mesenchymal cells in your bone marrow make more blood cells for your body to use, since red blood cells can't divide once mature. But red blood cells do eventually wear out and have to be removed from the blood.
Okay, so now I understand more about these stem cells. But why are they so important for research in medicine?
There are actually an excess of fields that can benefit from the use of stem cell research. Research is currently being conducted trying to utilize the regenerative properties of stem cells to stimulate regeneration of lost limbs or damaged organs. Being that stem cells are able to generate practically any somatic cell, and the body naturally distributes them to help repair organs, scientists are working to try to translate this function of stem cells into other tissues, such as bone or muscle. Currently, there is a lot of effort being put into the regenerative properties in organ transplantation. On average, nearly 20 people on the transplant waitlist die while waiting for an acceptable transplant. We have many artificial organs nowadays, but they are very expensive and often aren't covered by insurance. There are a lot of technicalities that can affect the success of a transplant organ that could be solved if we discover how to use stem cells to create or repair these organs. Imagine being able to grow a liver for a patient needing a transplant using that patient's own cells. there would be very little fear of rejection, which is all too common in regular transplants. The only hang-up with creating new organs from stem cells is that organs are complicated tissue systems that have a ridiculous number of cells involved to function properly.
I read an article about stem cells being able to help correct diabetes. It was really cool because apparently the stem cells could be injected into the pancreas and help produce more insulin for the body. Type 1 diabetes is due to the fact that the body cannot properly produce insulin, and this article claimed that stem cells have the potential to be able to cure these patients.
That's really cool. What else have you read about while doing research?
There was this one paper about using stem cells to be able to test the effectiveness of certain drugs and their long-term effects on the body. If they could figure out the exact triggers that cause stem cells to differentiate, as well as how to store them without letting them differentiate randomly, then this would allow the pharmaceutical industry to better understand how their drug affects every cell in the body by studying the effects on stem cells.
That's a great way to ensure that all of the side effects are understood before the drug is released. It really sounds like you're understanding everything better now.
But if stem cells are so useful and have so many different benefits, I don't understand how they can be so controversial. This is supposed to be a debate, and I have no idea what kind of questions will be asked.
Well, as beneficial as stem cells can be, there are a lot of setbacks. The most controversial of the stem cells are the ones that are obtained from the embryo.
Don't tell me they pulled the embryonic stem cells from the womb.
Actually, that's not the case at all. The embryonic stem cells are almost always recycled from in vitro fertilized eggs that would otherwise be discarded. So the embryos would not have been allowed to develop anyway, as they were fertilized in a lab setting and only the embryo chosen by the parents was put into a human surrogate. Therein lies a different ethical debate altogether, but it's unrelated to stem cells. But this common misconception is why there is such a stigma against research on embryonic stem cells, even though they are the best studied type of stem cell.
Well, that makes sense. But if it's not true that the stem cells are being harvested in an immoral way, then I still don't understand where the real issues with it lie.
Well, the main question of the debate surrounding stem cells really is whether or not our duty to alleviate human suffering through modern medicine is more important than our duty to respect human life.
I don't even know how you could question its medical benefits or deny its ability to alleviate human suffering. Obviously, all of the research we just described seems like it would be awesome for medicine.
Be careful, Alex. Have you considered that even though the cells come from embryos in a lab, that it means destroying the embryo anyway? Most of the ethical concern with embryonic stem cells follows the same debate over abortions, fertility clinics, and the disposal of fertilized human zygotes. Many people who oppose embryonic stem cell research, like those who are pro-life in the abortion debate, take the stance that these zygotes would, under normal conditions, develop into humans. Unlike a human, though, a zygote cannot give consent to the researchers, so many people feel like it violates the rights of a future human. This is why embryonic stem cell research is so touchy. It falls very close to the pro-life, pro-choice debate.
Oh, you're right. Well, I mean, baby killing is wrong. Now I don't know what to think.
Well, one thing someone in your debate might say in response to that argument is that the embryo has not yet developed any human characteristics, even at the cellular differentiation level, so therefore it has no human life qualities. In addition, many lab-grown embryos are thrown away each year because hospitals purposely create an excess amount. So some would argue that using the cells towards the benefit of human medicine is actually less wasteful and a more moral use.
I can see that. And I guess since other types of stem cells aren't as potent or have more side effects and technical difficulties, it would be difficult to just use another type of stem cell and avoid all this controversial mess.
Exactly. Now you're really catching on.
Well, what does the government have to say about this issue? Surely an ethical concern of this kind of scale hasn't gone unnoticed by the legal system.
Nope, you're right, it hasn't. In 2001, President Bush passed a moratorium prohibiting the use of federal funds on most stem cell research, while still allowing for research done in the private sector and done using state funding. His policy allowed for the continuation of research on existing cell lines, while disbarring funds to be put towards developing new cell lines, which was a huge hit to the stem cell research across the country. However, in 2009, President Obama overturned Bush's previous executive order to remove federal barriers from stem cell research on new cell lines, and though it was heavily challenged in the federal court system, the judges ruled to uphold Obama's policy and allow more stem cell research to continue. Currently, though, there is no national policy regarding whether or not stem cell research should be done and if funds should be allowed to support stem cell research. Laws governing stem cell research and funding are decided on a state-to-state basis, which means some states, like California, both allow for and encourage stem cell research in both the public and private sector, and allow for state funds to be put towards such research, while others, like Indiana, entirely prohibit the practice of stem cell research or human reproductive and therapeutic cloning. A majority of states, though, have in-between laws, creating some middle ground between the two, such as allowing the research using some types of stem cells, but not others, or only allowing for research in the private sector without providing them with state funds.
Wow, so even the states don't agree on stem cell use. I imagine that would cause conflict within the legal system. That really does just go to show that this issue is much more controversial than I once thought.
You've got that right. Well, I don't know what you think, but to me it seems like you've really got the gist of this stem cell thing now. Do you feel like you're ready?
Thanks, Erin. I'm really glad we had this conversation. I feel much more prepared now for this debate, and perfect timing too, because it's time for me to go.
No problem, Alex. I'm happy to help, and actually, I have to get going too. Good luck to you!